Hello, everyone. Welcome to today's final session of EscarpCon, day one. We're talking about fermentation innovation today. We've got a couple more days of content for you guys to learn a little bit more about fermentation and yeast in beer. I'm Richard, one of the co-founders of Escarpment Labs. I'm going to be the moderator for this webinar. And in this webinar, our topic is the history and future of lager yeasts. And we've got our special guest today, Kristoffer Krogerus. Kristoffer is a researcher at the VTT Technical Institute in Finland. They do a lot of cool stuff with brewing technology, including yeast. He's working in the lab of Brian Gibson, who they've done a lot of fantastic research on brewing yeasts and developing new brewing yeasts. And Kristoffer did his PhD on lager yeast, especially with regard to creating new hybrid lager yeasts and understanding these things a little better. And in the meantime, he's also done a whole bunch of other cool stuff. A paper on diastatic yeast that came out last year that's really helped the craft beer industry, and then also helped us out at Escarpment Labs with some of our research with Kvike yeast as well. So this guy's doing a lot of cool stuff. I'm really, really excited to bring him on here for this webinar series and tell us a little bit about the special story of lager yeast. For those that are joining us live, please feel free to start a discussion. We love getting questions. We're going to try to answer all of your questions at the end. I'm going to monitor the discussion and keep that conversation going as well. And then for anyone who's tuning in after the live cast, thanks for checking this out. Just want to mention to anyone that we do try to make these available on YouTube after the fact. So if you do miss any of the webinars, you should be able to access it any time. Without further ado, I want to give Kristoffer the opportunity to tell us about the history and future of lager yeasts.
Thank you very much, Richard, for the kind introduction. Hope everyone can hear me. So as Richard said, my name is Kristoffer. I work at VTT, Technical Research Center in Finland. We're a small group of maybe 6 people working with brewing yeast. And during my PhD, which I did 5 years ago, or started 5 years ago, finished this or last year, I focused on lager yeast and creating new lager yeast. And today I'll talk to you about, or give you a brief overview of what we know about lager yeast today and what we might expect in the future. And just a disclaimer, this webinar will be quite science heavy, so I hope you will be able to follow the more complex themes. And also, I'm not a historian, I'm a scientist, so please correct me in the questions or comments if I say something stupid about the history of lager yeast or anything like that. So yes, I'll try to share my slides here. Yeah, you can see them.
We can see them.
Yeah, great. Okay, so first of all, why are we interested in lager yeast or lager beer? Well, while the craft beer industry has exploded, grown tremendously, lager is still the most popular beer style worldwide, consumed and produced by far in the most volumes. And what's special about lager yeast is, of course, the relatively clean flavor profile. So it's neutral, easy to drink, and this is usually accomplished by cold fermentation. And lager is usually made with lager yeast, so Saccharomyces pastorianus. Not all lager beer is, of course, made with lager yeast, but in general, lager is made with lager yeast. What's special about lager yeast is that it's actually a hybrid between 2 different yeast species. So one of the parents is Saccharomyces cerevisiae, so what you may know as brewer's yeast or ale yeast or baker's yeast. The other parent was unknown for quite a long time, but in 2011, a yeast species called Saccharomyces eubayanus was discovered, and it turned out that this yeast species was the other parent of lager yeast. So it was already in the 1980s that researchers found out that lager yeast is a hybrid, but it took almost 30 years to find this other parent. And generally speaking, so this is a very simplified view of things, but in general, Saccharomyces cerevisiae strains, at least the brewing strains, they're good at fermenting wort, good at using the sugars in wort, but most strains are usually cold sensitive. Saccharomyces eubayanus, on the other hand, is quite bad at fermenting the sugars in wort. For example, isn't able to use maltotriose, but the strains are cold tolerant, so they can grow in the cold. And then the hybrid of the two, lager yeast, inherits the good properties of both parent strains. So it's lager strains are usually good at fermenting wort, and they're also cold tolerant. And just so you guys get a better idea of what a hybrid between two species actually is, there are examples from the animal and plant kingdoms as well. You might have heard of a liger or a zonkey, and many plants are also hybrids. So, for example, peppermint, and then most of the citrus fruit varieties are actually hybrids between different species. And hybrids, or these Saccharomyces hybrids, are also found in other fermented beverages, not only lager beer. So, for example, there have been hybrids found between Saccharomyces cerevisiae and another Saccharomyces species called Kudryavtsevi, this species here. So these have been found in many of the Belgian Trappist beers and spontaneously fermented beers. And then in wine and cider fermentations, you sometimes find hybrids between Saccharomyces eubayanus and Saccharomyces euvarum, and then Saccharomyces cerevisiae and Saccharomyces euvarum. So hybrids between different species in this Saccharomyces genus they're quite commonly found. But what's interesting is that these hybrids are usually found only in these man-made or like human-associated environments. So they're very rarely found in the wild. So these hybrids are usually associated with domestication. So if we go back to lager yeast and this their parent species, Saccharomyces eubayanus, as I mentioned, it was discovered in Patagonia in 2011. And since then, it has been found almost everywhere across the globe, except, well, not in Africa and surprisingly not in Europe yet. But it's very common in South America. It's been found in North America, in China and Tibet, and even in New Zealand. And the strains that have been found in North America and Tibet, the Holarctic strains, so these are the strains that are most close, genetically speaking, most close to the lager yeast genome. And there have actually been there's some evidence that Saccharomyces eubayanus might be present in Europe, because there was a study here came out in 2018 where they had looked at fungal or Saccharomyces, looked for the DNA in these forest samples, and they found traces of Saccharomyces eubayanus DNA in soil samples that were taken in the Italian Alps. So it is possible that Saccharomyces eubayanus is also found in Europe. It just hasn't been found yet, even though I know there are a lot of people looking for it. And why are we, or why would, and why are we, or why would we expect it to be found in Europe? Well, we'll be coming to that soon. But lager beer, or as you probably know, lager beer has its roots in Europe and in Germany. So it's very possible that eubayanus has at least once been found here, if it's not still just not found. So Saccharomyces eubayanus is not really suitable for traditional lager beer brewing on its own, but you can make a beer with it at least. So as I mentioned, isn't able to use maltodextrin, so that means you will end up with quite a low attenuation. So usually about, of course, depends on your mashing scheme, but maybe about 70% attenuation. And I guess one of the biggest disadvantages is that it produces phenolic off flavors. So for vinyl guaiacol, and this is something that is, of course, common in all wild yeast. So lager beer, of course, you don't want any phenolic off flavors in that. So in that sense, it's not really suitable for lager brewing. But it is cold tolerant, and it produces quite a lot of nice flavor active esters. And I don't know if any of you had the chance to taste this beer, but Heineken came out with a beer brewed with Saccharomyces eubayanus, the strain that was originally discovered in Patagonia. I don't know if they actually produce it anymore, but it's pictured here, this H41 beer. And I know that some craft brewers, or at least I think that some craft brewers in South America have produced some beers with Saccharomyces eubayanus, but someone will have to correct me if I'm wrong. And here's just to the right an example of a fermentation with lager beer, two lager yeast strains, so in blue and purple. And then in green we have Saccharomyces eubayanus. So you can see that it ferments quite slowly, and it doesn't reach the same attenuation level as the traditional lager yeast. But then again, it grows much better than Saccharomyces cerevisiae at cold temperatures. So if we start to think about the origins of lager yeast, we know that it was traditionally carried out at low temperatures, or is traditionally carried out at low temperatures. And at least since the 15th century, possibly earlier in Central Europe, and the roots are in Bavaria and Franconia, what is Germany today. And there are a couple of things that might have contributed to lager yeast emerging. So we don't know much about the details, might not ever find out, but there are certain things that, certain factors that might have promoted this hybrid from emerging. So we know that in the 16th century, in Bavaria, the Bavarian purity law was passed, and a bit later, brewing was banned in the summer months. So this was because the beer fermented in the cold rock cellars stored in the cellars usually was of better quality. So these laws were introduced because of that. And what this sort of can cause is that there was a pressure on the brewers to brew at lower temperatures. And this also, of course, simultaneously put the pressure on the yeast to ferment at lower temperatures. So these kind of conditions could have led to the evolution of a cold tolerant yeast, such as lager yeast. And we also know that the Saccharomyces cerevisiae parent of lager yeast is most likely or is a brewing strain, a beer one strain that is closely related to the Belgium and German strains. So you might have seen these sort of family trees of brewing yeast, and you know that the brewing strains group together in their own lineage. Let's see if I can get the mouse cursor here. So down here we have different beer strains, brewing strains. And here in green, light green here, we have the group with the Belgian and German beer one strains. So next to this branch here, we have the lager strains here. So the ale parent of the lager strains is most closely related to these Belgian and German Saccharomyces cerevisiae strains. And what this means, or what this supports, is that lager yeast has a Central European origin. And this split here is predicted to have occurred sometime in the mid-16th century. This was calculated in the reference that you see down in the right-hand corner, the recent paper from the Verstrepen lab. So you can look that up for the calculations and that. But this sort of makes sense, or fits quite well with the hypothesis that the lager yeast has emerged in Bavaria during the 16th century. So how did lager yeast then emerge? So there are several possibilities. One possibility is that brewers have made beer with whatever sort of yeast they were, or they of course didn't know about yeast as a microorganism then, but they were probably reusing sort of sedimented yeast, or reusing the barrels. And if a wild Saccharomyces eubayanus contaminant got into the fermentation, you might have a spontaneous hybridization with a Saccharomyces cerevisiae ale strain. And so this is actually, I mean, if you have a fermentation, if you mix two yeast in a fermentation, it's very likely that you will end up with a hybrid somewhere there in the population. Usually when we've been creating new hybrids in the lab, you might have a hybridization frequency of 1 in 100 million or something like that, or 1 in a billion. So it is quite likely that if you have two populations of yeast, that you might have some hybrids emerging. And then the conditions could have then favored the growth of the hybrid. The cold temperature could have favored it, and slowly over time, this hybrid could have outcompeted the parent strains. And why is it then likely that the hybrid would outcompete the parent strains? Well, there's an interesting phenomena with hybrids that's called heterosis, or hybrid vigor. And this is something that's often observed in hybrids, not only yeast hybrids, but all other kinds of hybrids as well. And this is something that's particularly has been exploited for a long time in agriculture. So, for example, in the picture to the right, you can see in the middle some maize or corn hybrids, and to the left and the right, you see the parent strains. So in agriculture, hybridization and heterosis has been used to improve crop yields, and you can see here that the size here of the maize is increased through the hybridization. So in the yeast hybrid work that we've been doing, we've also seen that hybrids often outperform their parent strains. So you can see here, we'll get back to this figure later in the webinar, but you can see here in the yellow and blue, we have the two parent strains, and in green, we have a couple of hybrids made between these two parent strains. And these hybrids are fermenting faster than either of the parent strains. So in this sense, if a hybrid formed at some point and the conditions favored the growth of the hybrid, it's very likely that it would have grown and outcompeted the parent strains at some point. Yeah, not much is, or the reasons behind heterosis is a bit, or it's a very complex phenomenon and not that well understood, but there are several factors that contribute to it. So when you actually form a hybrid between two different, for example, yeast cells, you combine the genomes of the two yeast species, then you increase the gene dosage. So, for example, you might double the amount of genes, and sort of genomes can cross-regulate, or you can see some cross-regulation between the two genomes. And sometimes even you can get these sort of new genes forming when these the genomes of the two parents sort of, or you can get these chimeric chromosomes and chimeric genes forming. So these are all possible mechanisms that sort of explain why hybrids would do better than the parent strains. But yeah, I want to emphasize that not all, or I mean, hybrids do not always outperform their parent strains. So it's not always true. But you very often see this heterosis effect. So between the traditional lager yeast that is available today, there's quite little diversity, especially compared to the ale strains or the Saccharomyces cerevisiae strains. So I don't now have the picture here, and I won't jump back in the slides, but if you remember that family tree that I showed you, those were all Saccharomyces cerevisiae strains, of course, from different origins, but there's a much wider diversity there compared to the lager yeast, which the genetic diversity between them is quite poor, as well as the phenotypic diversity. So we know that all the lager yeasts that are available today, they belong to one of two groups or lineages. So the sauce, or the group 1, or the Froberg, or group 2 yeasts. And there's been a bit of debate about if these are from two different hybridizations or a single hybridization event, but it looks like they're from a single hybridization event, and we'll get to that in a while. So in general, the sauce yeast, or the group 1 yeast, they are a bit less stress tolerant, but they are usually a bit more cold tolerant. And the Saccharomyces eubayanus genome sort of dominates in them, so they have more of the Saccharomyces eubayanus parent than the Saccharomyces cerevisiae parent in them. And one example of a sauce strain is Unterhefe number 1. So this was the strain that Emil Hansen isolated in Carlsberg, or the first pure yeast cultures. But it's quite interesting that these sauce strains are actually really rare nowadays in the commercial brewing culture. So there's been a couple of these recent genome sequencing papers where these commercially available lager yeast from, for example, White Labs and Yeasts have been sequenced, and there hasn't been a single sauce strain among these commercially available brewing strains. So they've all been Froberg strains, or group 2 strains. And these group 2 strains, so they usually have a bit more stress tolerance, but are not as cold tolerant. And here the amount of DNA from the parent strains is quite equal, but there's slightly more of the Saccharomyces cerevisiae genome in them. And a classic example is the Weihenstephan W3470 strain. But yeah, as I just mentioned, it's almost certain that if you buy a commercial lager strain, it's going to be a Froberg strain. These sauce strains can be found from various yeast culture collections. And I think that Richard would have to correct me, but I think that escarpment has a sauce strain, or at least has produced a sauce strain here recently. But I don't know if they're going to sell it continually. So if we look at the phenotypic traits of these two groups, so the blue, in blue we have the sauce strains, and in yellow the Froberg strains. So as I mentioned, the sauce strains are usually a bit more cold tolerant. So here you can see growth at 12 degrees, so a typical lager temperature. So the sauce strains are growing a bit better than the Froberg strains at this temperature. But then if we grow in the presence of 7% ethanol, you see that the sauce strains are not really growing at all, but the Froberg strains are doing okay. So the Froberg strains seem to have a bit better stress tolerance. And then one thing that, or for a while, it was sort of assumed that these sauce strains are unable to use maltotriose, but it's now been shown that most actually do. There are a couple of sauce strains that don't use maltotriose, and then a couple that use it really well. So that's sort of a misconception that these sauce strains wouldn't be able to use maltotriose. So there's not that much difference here. And the same here here's an example of isoamyl acetates production. So isoamyl acetate is the ester that's responsible for the banana aroma in beer. So there's quite little difference in the production of this ester between the sauce and the Froberg strains. In this picture here, you see the amount of chromosomes from both of the parent strains in the two groups. So the dots in red are different sauce strains, and the dots in blue are different Froberg strains. And on the x-axis, you have chromosomes that the strain has inherited from the Saccharomyces cerevisiae parent, and on the y-axis, the eubayanus chromosomes. So you can see that the sauce strains have a lot more of the eubayanus chromosomes than the cerevisiae strains, cerevisiae chromosomes, and the Froberg strains have quite equal amounts of both chromosomes, slightly more of the cerevisiae chromosomes. But what is quite interesting is that there's quite a lot of diversity here in the chromosome copy numbers. And it's very likely that while there's not that much difference in on the DNA sequence level, it could be that this phenotypic diversity that we see between the different lager strains is a result of diversity here in the copy numbers. So there's been some research showing that if you have increased copy numbers of chromosomes with certain genes, for example, related to diacetyl formation, then those genes will be expressed at a higher level and you will get more diacetyl formation. So it's very likely that the phenotypic diversity that we see between lager strains is a result of this diversity in the chromosome copy numbers. And hybrids in general, and also lager hybrids, they can be quite unstable genetically. So especially in these strains that have a very large amount of chromosomes. So in general, yeast or Saccharomyces yeast, they're diploid, which means they have two copies of each chromosome. In total, that would be 32 chromosomes. But these lager strains can have up to 80 chromosomes in total. So usually these higher ploidies, or it's called ploidy is the measure of these amount of chromosomes. So the higher the ploidy, the more unstable usually these strains are. And it's actually, I mean, if you take, there's been some research showing that even if you take samples from a yeast culture of a lager strain that you have taken from your frozen stock, if you randomly sort of look at different isolates from the same yeast culture, there might be differences in the chromosome copy numbers there. So these strains can be quite unstable. So here we come back to the Saccharomyces cerevisiae parent, and so this is the beer one group, and you see here on the x-axis, this is sort of a sort of predicted timeline of what's been happening. So it's predicted that the split here between the Saaz and the Froberg yeast has happened maybe in the beginning of the 18th century, and compared to the cerevisiae strains, there's sort of a bottleneck here quite late, and when these pure yeast cultures were isolated, probably then distributed around the world, especially here in Europe, and that has sort of limited the genetic diversity between these lager yeasts compared to the cerevisiae yeast. So as I mentioned already, it's quite likely that the hybridization has only happened once. So in early research, it was predicted that there would be two different hybridizations, as depicted here in the A panel. So we have and then this was based on the fact that they had such different chromosome copy numbers. So the Saaz strains appear to be diploid for the eubayanus parent and haploid for the cerevisiae parent, and the Froberg yeast diploid for both of the parents. So yeah, this is related to the copy numbers that I showed you a couple of slides ago. So the hypothesis was that for the Saaz yeast, you would have had a haploid cerevisiae strain mating with a diploid eubayanus strain, and then for the Froberg yeast, you would have the diploid cerevisiae strain and a diploid eubayanus strain. But when when people have looked at the DNA sequence of these, or sequences of different Froberg and Saaz strains, there's a very high similarity in both the cerevisiae and the eubayanus sequences in both groups. So that's why it seemed likely that it would have been just a single hybridization event, or why people started looking into that. And there is a bit of a heterozygosity in this cerevisiae genome. So that means that in these two copies of the cerevisiae chromosomes, there's a slight difference between those as well. So one hypothesis was that we would have first had this sort of a lager yeast ancestor, which would have been between a haploid cerevisiae strain and a diploid eubayanus strain, and then this would have sort of formed the Saaz strains, and this would have sort of mated again with another haploid ale strain to form this Froberg group. And then the last scenario, which is maybe the most likely, is that we have a diploid cerevisiae and a diploid eubayanus mating to form this lager yeast ancestor, and then this Froberg Saaz group would just have lost a part of the cerevisiae genome. Some more evidenced the fact that there's probably only one hybridization event is that there are in this lager yeast genome, there are a lot of chromosomes that are chimeric. So you have a part of the chromosome is a eubayanus chromosome and part is a cerevisiae chromosome. And so there are many of these chimeric chromosomes, and many of these chimeric chromosomes are found in both the Froberg and the Sass yeast. And in many, and the sort of break point here where the two chromosomes merge, they happen in the same place, exactly the same place there in the sequence, in both the Lager and the Froberg and the Sass strains. So it's very unlikely that these sort of same breakpoints or chimerization events would happen if there were two different hybridization events. So this sort of supports that there's only a single hybridization event. So to conclude sort of my first half of this webinar, we know that there are two subgroups of lager yeast, so the Sass and the Froberg. And unfortunately, if you buy a commercial culture of lager yeast, it's almost guaranteed to be a Froberg strain. There's not many Sass strains available commercially. Maybe we'll see more of them in the future. And so they have a bit different phenotypic properties. So these Sass strains with a bit more of the eubayanus genome in them are a bit more cold tolerant, while the Froberg strains, which have a bit more of the cerevisiae genome, are a bit more stress tolerant in general. And yeah, that brings me on to the next or the second section of my presentation, and this will focus on strain development. So since there is limited diversity among the lager yeasts that are available today, the question is, how do we improve the diversity, or how do we improve the lager yeast strains that are available today? So there are, of course, a number of different traits that are interesting for the brewer. I guess it depends exactly on what type of beer you're brewing and what kind of brewing process you have. But sort of some of the general things that one could be interested in is improved stress tolerance, or perhaps the production of or enhanced production of aroma compounds that are desirable, or decreased production of aroma compounds that are undesirable. So this is what I'll focus on most here. So I'm going to talk about 4 different sort of strategies or methods that can be used for developing strains. So the first one is random mutagenesis. This is when you use a physical or a chemical mutagen to cause mutations. And as the name indicates, this happens at random, so you're not really sure what you're going to get. Then I'm going to talk about adaptive evolution. So this is when you're trying to replicate natural evolution by applying some kind of a selective pressure to enhance a trait that you're interested in. Then I'm going to talk about yeast breeding or hybridization. So this is the topic that's most familiar to me, because this is what I did my PhD thesis on, specifically creating new lager yeast. And finally, I'm going to talk briefly about genetic engineering and how that can be used to improve brewing strains. So if we begin with random mutagenesis. As I mentioned, these mutations are randomly introduced. Usually people use ultraviolet light. So ultraviolet light causes damage in the DNA sequence. And there are also different chemicals that can be used that are sort of a bit more specific in the type of change they cause. But it's still completely random where in the genome these mutations would occur. So one of the advantages is that there's no genetic engineering involved. So that's of course still a bit taboo topic, this genetic or the use of genetically modified yeasts in the food industry. So it's at least not in Europe allowed yet, but in North America, I've understood that it's sort of opening up a bit, and these strains are more available. You can generate quite a lot of diversity with the mutagenesis, but the problem is that you're not really sure where these mutations are occurring. So you need to somehow be able to screen and select the good mutants from the bad ones. And because this is completely random, you might get a lot of unwanted changes in the phenotypes as well. So I'm going to give you here a couple of examples. So the first case here is if we want to decrease the formation of diacetyl. And this is, of course, an important topic for lager brewing because this can usually be a problem there. So diacetyl is formed when the yeast cell is producing their own valine or leucine. So these are amino acids. The yeast can take them up from the wort as well, but usually the yeast isn't able to absorb enough or quickly enough from the wort. It has to create its own valine, and while it's producing its own valine, it's producing alpha acetolactate, and this compound gets converted into diacetyl. And this first step here in the process, it's catalyzed by an enzyme, or the gene that codes for the enzyme is ILV2. And there's a chemical called sulfometronmethyl, or there are actually many different chemicals that inhibit this enzyme. And this is toxic to the yeast, or if there's no valine available in the growth media, then the yeast can't produce their own valine because this gene or this enzyme is inhibited by this chemical. But there are when you sort of treat or when you grow yeast in the presence of this compound, some mutants are resistant to this compound, and it's been shown that these mutants usually have mutations in this ILV2 gene. So if you perform random mutagenesis and then grow yeast in the presence of this chemical here, then the yeast that are sort of growing well, that are resistant to this chemical, they most likely then have mutations in this gene, and this can cause less of this precursor to be formed and less diacetyl as well. So here's just an example of a wild type strain and a mutant, a resistant mutant that has been derived from this strain. So when final attenuation has been reached, there's almost 3 times less or 3 times more diacetyl in the wild type compared to the mutant. So here, in the beer made with the mutant, the diacetyl level is already below the threshold at the final attenuation, while the wild type might need a day or two more of maturation. So here's another example where you might want to increase the amount of isoamyl acetate. So this was the ester that's responsible for the banana aroma. So here we can use a compound, I won't try to pronounce it, but this compound here, which is also a toxic chemical for the yeast, because the yeast sort of thinks it's leucine, the amino acid. So it's a like fluorinated version of leucine. So while it's toxic to the, or in the same way, well, if there's no leucine present in the growth media, then this compound will inhibit a G enzyme that's encoded by this Leo4 gene. It's this last step here. But then there are mutants that are, again, resistant to this chemical, and these have mutations again in this gene. And in this case, you actually see a doubling of the isoamyl acetate produced. So here, the wild type strain, or in the resistant mutant produced twice as much isoamyl acetate as the wild type strain. So then we will move on to adaptive evolution. And as I mentioned here, we're trying to replicate or speed up the natural evolution that happens, but in the lab setting. And here we exploit the genetic diversity that's present in a population, and then we apply, or we cultivate the yeast in such an environment that it selects for a certain trait that we're interested in. So this can be quite difficult in many cases. So if you're, for example, interested in increasing stress tolerance, then this is quite easy to do, because then you just grow the yeast in presence of that particular stress that you're interested in, because then you sort of naturally select for the strains or the mutants that are more stress tolerant or growing better in the presence of that stress. But then if you're interested in, for example, improving aroma, you want to produce more isoamyl acetate, for example, then it can be a bit more difficult, because you need some kind of growth conditions that will promote the growth of a strain that produces more aroma. And there, it might be difficult to sort of link the phenotype that you're interested in with sort of a selective environment. So again, the advantages is that there's no genetic engineering involved, and it's quite effective at enhancing desired phenotypes as long as you are able to select for it. So as with the mutagenesis, you need to be able to screen and select the trait that you're interested in, and it can also be quite time consuming. So you might have to grow the yeast for several hundred generations, so that can translate into many weeks before you see a desired outcome. So here are some examples from my own work. So I was interested in increasing the ethanol tolerance of some of the hybrids that I had created. So what I did was grow the yeast in a growth media that contained 10% ethanol. So 10% ethanol, the growth is already inhibited quite a lot for most yeast strains and brewing strains. So in 10% ethanol, you would get quite poor growth. So I repeatedly grew yeast for 30 weeks. So I grew them for 1 week in media containing 10% ethanol, and then transferred to a fresh media after 1 week. And I did this for 30 weeks, so 29 different transfers. And you can see here in the figure here, on the x-axis, you have the different weeks, or the different batch fermentations. And on the y-axis, you have the optical density of the culture. So this is sort of a measure of how much the yeast has grown. So you can see that there's sort of a increasing linear trend here, that the longer the yeast were exposed to this environment, the better they had grown after that 1 week period in the media. When we then selected some sort of adapted variants from the adaptation and compared them with the original unevolved strain, we also saw that in wort they outperformed the original unevolved strain. So here you have I looked at a triploid hybrid and a tetraploid hybrid, and in black you see the unevolved strains, how they fermented in wort. So this was 15 Plato wort. And the blue and red lines there are then how two different adapted variants from both of the original hybrids fermented compared to the original strain. So here you see that the adapted strains fermented quite a lot faster than the original unevolved strain. And when we looked at ethanol tolerance, we also, in most cases, saw an increase in ethanol tolerance, which is sort of expected because that's what we selected for. You can see here in this graph, you see how much ethanol was produced from a really high gravity media. So this is 35% maltose, so it sort of translates into 35 Plato. And so the adapted strains fermented better than the original strain. And then here you see how they grow on agar with 9% and 11% ethanol, and you see that the adapted strains are growing better than the wild type strain. So a stress like ethanol is usually quite easy to select for. So these adaptive evolution studies are usually quite effective for increasing stress tolerance. And here's another example of increasing maltotriose consumption. So this was with a lager strain, and what the authors did was grow the lager strain in a continuous carbon-limited culture, and the media that they used had a lot of maltotriose in it. So it wasn't pure maltotriose, if I remember correctly. There was a sort of a blend of the wort sugars, but the concentration of maltotriose was much higher than you would expect in a wort. So by sort of continuously growing this lager strain in an environment with a lot of maltotriose, they were selecting for strains that were growing or consuming maltotriose better. And when they compared the original strain here, in white, with the adapted strains, here you see the amount of maltotriose consumed during fermentation. So you see that the adapted strains were consuming maltotriose better than the original strain. So this brings me to yeast breeding or hybridization. So here I will talk a bit about the work that I did during my PhD thesis. So what's nice about yeast breeding is that you're able to combine properties from many different strains into one. And again, there's no genetic engineering involved in most cases you can do it without genetic engineering. And as I showed you previously, these hybrids you can often see a heterosis effect. So not only are you combining properties from many different strains, you are potentially enhancing them, making a strain with, for example, better fermentation or more production of aroma. Disadvantages is that sometimes it can be a bit unpredictable what kind of phenotypes you will get, and not all strains are suitable for hybridization, or especially many of these brewing strains are quite difficult to work with or create hybrids with. But it's not impossible, but it can be quite challenging. So why we started this project, or sort of the idea that we had in the beginning of my PhD thesis, was that could we increase the diversity of lager yeast by creating new hybrids? So this was when Saccharomyces eubayanus had quite recently been discovered, and so we wanted to try to create new lager yeast by hybridizing different ale strains of cerevisiae with Saccharomyces eubayanus, sort of replicating the hybridization that had happened probably then in Bavaria in the 16th century. So just so you get a better idea of how this is actually done, here I'll just briefly go through the process. So it's actually a very simple process, or a simple thing to do. As I mentioned, when you mix two strains in a culture, it's very likely that you will have some hybrids forming at random. But the big challenge is, how do we pick out that one hybrid out of 1 billion other cells? There are a couple of different ways that this can be done. The sort of approach that I took was that I sort of tagged these parent strains with auxotrophic markers. So this means that you sort of select mutants of the parent strains that are not able to produce different amino acids. So, for example, this eubayanus parent strain, we selected a strain that wasn't able to produce its own lysine. And what this means is that if you try to grow the strain in a media that doesn't contain lysine, then you won't get any growth at all. And for the cerevisiae parent, we selected these uracil auxotrophs. So what we then simply did was mix the two parent strains and grew them in standard growth media. And then if you plate them or spread out the yeast onto a agar plate that doesn't contain lysine and doesn't contain uracil, then only the hybrids will be able to grow. So the parent strains won't be able to grow because they can't produce these. Well, uracil isn't an amino acid, but it can't produce lysine and uracil themselves. But the hybrids can produce them themselves because they get the functional lysine and uracil synthesis genes from the opposite parent. So this is sort of quite a easy way to create and select hybrids. And then you can just use PCR to make sure that you have DNA from both parent strains in the hybrid. So here is the same figure that you saw earlier in the presentation. So when we compared some of the hybrids that we had created between a cerevisiae ale strain here in yellow and a Saccharomyces eubayanus strain, we saw that the hybrids were fermenting faster than either of the parent strains, and also reached higher attenuation levels. And also when we looked at the aroma compounds, so here you see ethyl hexanoate, which has sort of an apple-like aroma, and isoamyl acetate, which again has a banana-like aroma. The dotted line here is the flavor threshold. So if the concentration is above this line, then on average, a person is expected to be able to tell that aroma. So the hybrids produced much higher levels of this ethyl hexanoate than either of the parents, and for isoamyl acetate, the highest levels were observed in the eubayanus parent, but the hybrids were not far behind. So in general, the beer produced with the hybrids was sort of more aroma rich. This slide here is probably quite complicated, or complex and complicated to look at and understand. So I'll try to sort of describe what's happening here. So this is a study from the Verstrepen lab. They were working on a similar project that I was at the same time as I was, and they focused more on aroma, or specifically on aroma diversity. And so they wanted to see if they could improve the aromatic diversity of lager yeast by creating these new hybrids. So here in this heat map here to the right, you see different hybrids mostly, and then some cerevisiae parents, different sauce type lager strains, Froberg type lager strains, eubayanus down here. So you can see that there's a lot of diversity. There are different aroma compounds here, the columns, and depending on if the color is blue or red, you have a low or a high concentration of these aroma compounds. So there's a lot of diversity. Some strains or hybrids are producing a lot of aroma, some are producing very little aroma. And then if you sort of do a principal component analysis of this data, you can sort of reduce the complexity of the data into only, here's 3 dimensions. You see that in the strains here in blue, dark blue and light blue, if I remember correctly, the dark blue strains are the Froberg lager yeast strains. In the light blue are the sauce type lager strains, and the black strains are newly created hybrids. So what we see here is that there's with the newly created hybrids, we see a sort of expanded diversity of aroma compared to what you get from just the traditional lager strains. And then in the beginning of the project, I was only creating hybrids between 2 different species. But I also wanted to see if I could create hybrids with more than 2 parent strains, because this would sort of increase the applications of hybridization. So here's an example, sorry, of some research that we did where we tried to create hybrids with 3 parent strains, all having different desirable properties. So we have parent strain 1, which is able to use maltotriose, but it produces 4-vinyl guaiacol. So it's POF positive, and it's also cold sensitive. Parent strain 2 is POF negative, so it doesn't produce 4-vinyl guaiacol. And parent strain 3 is cold tolerant. So the way in which one can create these triple hybrids is that one first produces a double hybrid, so a hybrid between 2 different strains. And then one takes this hybrid and one mates it with the 3rd parent strain. So usually hybrids are sterile. So you might have heard that these animal hybrids, such as ligers or mules, they're sterile, so they can't get any offspring of their own. So yeast hybrids are also usually sterile. But what we figured out was that if you create tetraploid hybrids, they're actually fertile, so you can sporulate them. And so it's not that important for you to know what a tetraploid hybrid is. It's just important that you know that you can sporulate them, because that means that you can easily mate them with another strain. So we created a double hybrid, sporulated it, and then we were able to mate these spores with the third parent strain to create a sort of triple hybrid. But the problem was that, yes, it was able to use maltotriose, and it was cold tolerant, but it still produced 4-vinyl guaiacol. And this is, of course, because 4-vinyl guaiacol is a dominant trait. So we know that 4-vinyl guaiacol or phenolic flavors are produced when the yeast has functional PAD1 and FDC1 genes. And since the triple hybrid sort of had gotten the functional genes from these two parent strains, this triple hybrid would also produce phenolic flavors. But we were able to get rid of this phenolic flavor trait by again sporulating this triple hybrid and selecting for sort of a spore that wasn't able to produce phenolic flavors. And this is because when you sporulate a yeast, you sort of get exchange or recombination of the genetic material. And since this triple hybrid also contains some of the non-functional genes from this second parent strain, some of the spores would contain that as well, and only that. So when we did fermentations with these different strains, we saw 4-vinyl guaiacol production in all strains except the original parent strain, which was POF negative, and then this T2 triple hybrid that was selected after sporulation. And this is sort of an important result because all the hybrids or newly created hybrids up until this point, they had always been POF positive. Because if you create a new hybrid with Saccharomyces eubayanus, because that parent strain is POF positive, then your hybrids will also always be POF positive. And this is, of course, something that you don't want when you're making lager beer. You don't want any phenolic flavors. So this was sort of quite important for us, this result, that we were able to create new lager hybrids that are POF negative, that don't produce any 4-vinyl guaiacol. And what was also quite nice was that this T2 triple hybrid was also the strain out of these 8 that fermented the best and consumed the most maltotriose. So not only did we produce a hybrid that was POF negative, but it was also one of the best performing ones in fermentation. So this brings me to the final part, genetic engineering. So this can be used to either remove unwanted traits or add traits, also novel traits that are not naturally found in the species. And this is quite interesting because then you can sort of create truly unique strains. And the advantage is, of course, that it's very precise. You know exactly where you are creating the modification in the genome. It's not like with mutagenesis that you're creating a lot of random mutations. You're usually able to target the mutation, especially to a specific place in the genome. But as was mentioned, the use of these genetically modified yeast is still a bit taboo. But I'm sure we'll see a change in the future, probably near future already. And I guess sort of a disadvantage is that complex pathways can be quite difficult to introduce. So if you're interested in the production of a certain novel aroma, it's usually not just sort of one gene that you have to insert. It's usually a long, complex pathway, and this can be quite challenging. So again, I'm going to show you a couple of examples. So if we go back to diacetyl, and we again want to decrease diacetyl formation, then one simple way of doing this is just delete the gene here that is responsible for the enzyme that catalyzes this step. Or actually, one can delete sort of a sub unit of this gene. So as I mentioned before, the enzyme here is encoded by ILV2, but then there's also a gene called ILV6, which produces a protein that sort of regulates how active this enzyme is. So by deleting this ILV6 gene, you're sort of inhibiting this reaction here. So less of the precursor is formed, and also less diacetyl. So here you see an example of the wild type lager strain. Here on the y-axis you have diacetyl concentration. So this is during fermentation. You have an initial increase in diacetyl, and then with maturation you get a decrease. And with the strain where you delete this ILV6 gene, you have a much lower concentration of diacetyl throughout the fermentation. And you see here is how the fermentation progressed, and there's not much difference between the wild type strain and the strain where this ILV6 is deleted. I guess this is can be one of the dangers that if you sort of delete a gene responsible, for example, for a certain aroma formation, then it is possible that you sort of affect some other properties, such as how the yeast ferments. Especially here in the case of diacetyl, when this is involved in production of a amino acid, then it might be that you sort of change how well the yeast ferments. And this is quite simple to do, especially with the new modern tools such as a CRISPR-Cas9. And then this is especially useful if you have a trait that is only controlled by a single gene. So this could, for example, be the phenolic off-flavor production. You can, just by deleting either this PAD1 or FDC1 gene, then you get rid of the whole phenolic off-flavor production. And similarly, in some of our work with diastatic yeast, if one deletes this STA1 gene, then you eliminate the phenotype completely. But this is a lot more complex if you have a trait that is controlled by many different genes. So, for example, ethanol tolerance, it's a lot more difficult to increase, for example, ethanol tolerance by deleting various genes, because there are so many different ones that affect ethanol tolerance. Then if we again bring up isoamyl acetate, and if we want to increase the formation of this ester, then it's quite simple to do. All you have to do is overexpress the ATF1 gene. So this ATF1 gene is the gene that encodes the enzyme that is responsible for the ester formation. So when these esters are formed, you have a reaction between a higher alcohol, in this case isoamyl alcohol, and then acetyl-CoA. So by overexpressing this ATF1 gene, you're producing more of this enzyme that catalyzes this reaction. And the more of this enzyme that is present, the more of the product will be formed. So how this is done is that in general, when you have a gene such as ATF1, in front of the gene there in the DNA sequence, you have a promoter, and this promoter controls how much of this gene is transcribed. So basically tells how active the gene is. But if you switch out this promoter to a promoter from a gene that is produced all the time, so for example, one of these central metabolism genes, for example, involved in glycolysis, if you switch out the promoter of this gene to it's called a constitutive promoter, then this gene will be active all the time, or will be transcribed all the time throughout fermentation, and you will get a much higher expression level. So in this way, you can create some strains that really produce a lot of isoamyl acetate. So here, by overexpressing ATF1, you can get a strain that produces almost 200 times more isoamyl acetate. And at these concentrations, I don't have any experience myself, but I can imagine that the beer is totally undrinkable, or smells and tastes only of banana. So it can be a bit sort of difficult to control sometimes, or you would have to probably experiment a bit with the promoter that is used to get the sort of palatable level of isoamyl acetate, if you would be interested in an approach like this. Then finally, I'm not going to go into details, but I'll just mention them that yeah, with genetic engineering, you can sort of introduce some novel flavors, non-native flavors. So there's been some studies where they've got a yeast to produce a raspberry aroma from this raspberry ketone, vanilla aroma by producing vanillin, cinnamon aroma by producing cinnamaldehyde, and then also producing these hop aromas or hop terpenes by producing linalool and geraniol. So you can really make some unique yeast strains in this way, but of course it's not just simply introducing the genes into the yeast. It will probably require a lot of optimization and things like that if it's a really complex pathway that is responsible for these production of these non-native aroma compounds. So that brings me to my conclusions here in this second part. So there are many different ways of improving your yeast strains, and they can broadly be divided into the non-GM strategies and the GM strategies. So if you don't want to have any genetic engineering, then you have to rely on the natural diversity, and you can, for example, use adaptive evolution or breeding or hybridization. For adaptive evolution, you will need to be able to select for the phenotype that you're interested in. This is easy to do if it's something that affects growth, especially stress tolerance is quite easy to improve by adaptive evolution. But then if you're sort of instead interested in combining properties of many different yeast strains, then hybridization or breeding is sort of the path to take. And yeah, in general, these non-GM methods will require good screening and good selection. Otherwise, they're not that effective. And then the GM methods, on the other hand, you can do some really precise modifications. But of course, there's still a bit, yeah, in some cases, it's not allowed by law, as in, for example, the European Union. to use these strains. In other cases, it's maybe more about consumer acceptance, getting consumers to accept the use of these genetically modified yeast in the brewery. So that brings me to my final slide. Thank you all for listening, and if you have any questions, I'll be happy to try to answer them. Can you hear me, Richard?
Yes, can you hear me?
Yeah, I can hear you now.
Okay, it would not let me bring my mic back online, but I'm back now. Thank you, Kristoffer, for the informative talk. I think it was a really good deep dive into lager yeast and how it came to be. Definitely no shortage of information about the genetics of lager yeast. I did see some people following along in the comments talking about ploidy, talking about the differences between Saaz and Saaz and Froberg strains, and a lot of fun stuff. So I'm glad that there are some people following along. And for those of you who maybe aren't so technical, I think the second half of the talk would be really cool. Regardless, just learning about all the ways that you can, in the lab, you can make yeast that make less diacetyl. You can make yeast that make more esters. Yeasts that are more tolerant to ethanol, ferment faster. There's a lot of really cool potential in in some of the lab work that's being done these days, and that's exciting. And, hopefully, we can start to see some of that roll out into the commercial side as well in the future. So thank you for the talk, Kristoffer. Super informative. We do have some time for some questions, and we do have a number of questions. We can go ahead and dive into that. The first question is, are there strains available to brewers which are part of Group 1 Saaz? Do you know of any modern breweries that are still using these type of strains for lagers? Especially interested in Czech breweries that may be using these strains.
I'm actually not that sure. I think that Carlsberg is still using, or the Carlsberg or the original Unterhefen one that was isolated by Emil Hansen. I think so. That's the type strain of the Group 1 yeast. You might have heard of Saccharomyces Carlsbergensis, as it was originally called, but it's now been renamed to Saccharomyces pastörianus. So I think that the Carlsberg are still using that strain, but I'm not, or like a derivative of that strain, but I'm not completely sure. And as I mentioned, otherwise these Saaz strains are quite difficult to get a hold of. I don't know if I know that you guys produced that one Saaz strain. Is that something that you'll continue to produce, or was that just a one-time experiment? Or
yeah, probably not. It's a bit of a pain in every respect. So from propagation through to the fermentation, we encountered numerous challenges. So one of the kind of interesting things is we use molasses in our propagation medium. It's very common for yeast propagation just because it's a really cheap and high quality source of nutrients for yeast. That yeast does not ferment in the presence of molasses. It would not, we'd get some biomass, but it wouldn't ferment the sugars whatsoever. So there's something there that's inhibitory to the Saaz yeast specifically. And then, through to fermentations, we had one promising, or a couple, promising lab scale ferments, but then scaling that up to the brewery size has been a big challenge. So we're still, I think if we were to proceed with that, we really need to nail down, exactly the right handling procedure, and make sure that whoever is getting it is able to follow that, just because it's a lot more challenging than the Frobergs. All of our commercial lager strains are Froberg yeasts, and those are the ones that are, by and large, the easy ones to use. There are still differences within that group. That's sort of been one of the confusing thing is people, think that Satz means Czech. But it's actually like, at Carlsberg yeast. And that there are some Czech yeasts that seem to have those traits, like slower fermentation and more like volatile sulfur and stuff like that. But those end up being Frobergs as well. So
but if someone is interested in getting hold of a sauce strain, then they can be ordered from various culture collections around the globe. Just Google yeast culture collection. I'm sure you'll find a lot of different ones to choose from. If not, then we can send from VTT's cultural collection, where we have many of them. But I guess one of the problems there is that usually these culture collections, they are the strains are only sold for research work. So if you're a brewery and want to use it for commercial brewing, then it can be a bit challenging to get a hold of these strains. But I mean, I don't have that much experience in that, but
you can license cultures from culture collections, and some of them are reasonable, if you're looking at a yearly licensing fee for commercialization. How do you think lager yeast came to European countries? I think we sort of covered that a little bit in.
I mean, there was in the beginning, after it was originally found in Patagonia, there were these hypotheses that it would have somehow, eubayanus would have somehow come over by trade or something like that, or lager yeast would have emerged there in South America and then been traveled back to Europe. But all signs point towards it forming in Europe, probably in Germany, and it's just that we haven't found eubayanus yet in Europe. Either it doesn't exist anymore, or then we've just been looking at the wrong places, or whatever. I'm not really sure, but I still hope that it will be found one day here. We've been looking for it ourselves, but haven't been successful. We thought that the Finnish climate would be perfect for this cold-tolerant yeast, but we have only been able to find Saccharomyces paradoxus in the wild, or a lot of that. It's quite common here in Finland, at least.
Same here as well. I know that there has been eubayanus found in Canada, but we haven't found any. It's usually paradoxus if we're looking at wild yeasts. So still a bit of a mystery where the heck this stuff is in Europe. Maybe the original environment is gone, right? There's been a lot of development, deforestation and development of agriculture. Maybe the habitat is destroyed.
Yeah, exactly.
Has anyone measured how many? So you talked about hybrids. Has anyone measured, obviously, how many generations it took for a hybrid to outcompete the original strains in a culture? So you said like hybrids show up in a 1 in 100 million, 1 in a billion kind of rate. Like how long does it take for one to become dominant?
Yeah, this is something that I've thought about myself, and sort of an experiment that I was thinking about, or I had an idea for an experiment to perform early in my thesis, but I never got the chance to do it. But I thought it would be really interesting to sort of follow the dynamics of a hybrid, or sort of you would mix, for example, equal amounts or in different amounts, the cerevisiae and eubayanus, and then you would have some way to monitor easily how much of the different cells are present. So I was thinking that you could sort of tag them with these fluorescent markers, so that one of the cerevisiae strains would, for example, fluoresce with some green fluorescent protein, and then the eubayanus with some yellow or red, or some different. And then you would sort of monitor the fluorescence of the different parent strains, and the hybrids would then fluoresce, or have produced both of these fluorescent proteins. So you could sort of easily, with flow cytometry, sort of count how many of the parent strains and how many of the hybrids there are, and then you would sort of follow, grow them in different conditions, different media, and sort of see if would the hybrids actually outcompete the parent strain. So is this hypothesis of a eubayanus contaminant plausible or not? But yeah, I unfortunately never got the time to do that, but maybe someone will perform that experiment in the future.
That's a great idea. The next question is, a lot of research suggests that yeast can lose the ability to efficiently ferment maltose after a few generations in glucose or fructose environments. Do you think that yeast losing the pressure toward efficient maltose consumption would cause them to lose the adaptation on such a short timeline? What's going on there with maltose consumption?
Well, yeah, it seems to be that if you sort of grow yeast on glucose, then it at least for a while loses the ability to grow on these more complex sugar sources. But this is usually not a genetic change. It's sort of some other adaptation that's happening. And I think that there was a paper from the Verstrepen lab again, where they had looked at this lag phase when you shift from glucose to maltose and glucose, and the shifting from these different sugars. And I think it's quite common that you if you grow a yeast on maltose, for example, for a long time, then it sort of adapts to using maltose, and you can, if you use them in a wort fermentation, there will be very little lag phase. But then if you transfer it back to glucose, even for a short while, then it completely loses this ability, and then you sort of have to get it back or adapt it back to maltose. And this is something that we've seen a lot when we've tried these different wild Saccharomyces strains. So usually they perform quite poorly in wort fermentations if you grow them, propagate them on glucose. So they usually have a very long lag phase before they start using maltose. But then if you sort of propagate them for a while on maltose, then this lag phase disappears. So I don't remember exactly the cause for this, why we see this, but yeah, I can see if I find the link to that paper, and I can post it in the comments or something like that, if Crowdcast allows that kind of thing. Or
I think you can.
Yeah.
Yeah, I mean, I guess it might come down to this phenomenon of catabolite repression, right? If glucose is, then it kind of represses a lot of this other machinery from being activated. So that might be what's going on.
Yeah, and definitely these maltose. So for the cell to be able to use maltose, it has to transport it into the cell through the cell membrane. So these transport proteins, the genes that encode them, they're usually repressed by glucose. So if there's glucose present in the media, these transmembrane proteins won't be produced. So the cell isn't able to take up maltose in that case.
Yeah. Did you work out which genes were altered, leading to the increased ethanol tolerance in those adapted yeasts?
We did look at that. Yeah, I chose to exclude that from this presentation here, but I can again get the link to the paper there. But we looked. So we had 4 different strains that we adapted. So one was this cerevisiae parent strain, and then we had 3 different hybrids that we had created ourselves, and they were all subjected to the same adaptation protocol. And we saw a lot of different genes that were affected, and some were affected in many different strains, or like we saw the same genes that were hit by mutations in many different strains. But in general, there was not really an obvious, there weren't like any obvious mutations in most cases that explain this. But what we saw that was quite interesting was that there was a lot of copy number changes in the chromosomes. So in many cases, after the adaptation, the adapted strains would have gained or increased amounts of certain chromosomes, and they had lost certain other ones, and there were a lot of common losses and gains. As ethanol tolerance is sort of a polygenic trait, so there's a lot of different genes that affect it. It's not, unfortunately, that simple to find like specific, or we weren't able to find, or only in a couple of strains there were genes that previously in other papers had been shown to directly influence ethanol tolerance. So we had a couple of those. I think one gene was UTH1. So if UTH1 is not functional, then you get better ethanol tolerance, for example.
Okay, what does that gene do?
Now I don't remember, unfortunately, but
okay, that's interesting. So I just have one final question before we cap this one off. So there's been a lot of great science on developing these new lager yeasts, by yourselves, but also by other groups. And I'm wondering, at what point are we going to start seeing some of these new hybrids enter the market? Like, at what point will these be available for purchase? It seems, there's a lot of cool science, but then we're not seeing these things leave the labs yet.
Yeah, that's a question I would also want to get answered. I'm sure that, or at least our lab, and I'm sure all the other labs that have been working on the same thing, they have hybrids ready to be sold if anyone is interested in trying them out. I guess one problem one thing that many have been sort of skeptical about is that these hybrids or newly created hybrids can sometimes be unstable, as I mentioned, so their genomes can quite easily change. And sort of this was also one of the things that why we wanted to do this adaptation process on some of our hybrids, was to sort of stabilize them a bit, so create these changes in the genome already, so that they won't happen spontaneously in the original hybrid. So I'm sure there are strains available if breweries or yeast labs are interested in them. I guess one thing that has also complicated things a bit regarding these lager hybrids is that many of the original hybrids that were created by the different labs, they were using this type strain of Saccharomyces eubayanus, so the original one that was discovered in Patagonia. But as these were brought from culture collections only for research work, we're not allowed to sort of commercialize them. And I know that Heineken, which then makes this H41 eubayanus beer, I think they have sort of exclusive rights to this type strain, or some of these originally discovered eubayanus strains. So any hybrids that would have been created with that strain can't really be used unless you, I guess, maybe can sort out some kind of agreement with Heineken, but I'm not sure how easy that would be. But since the original discovery of eubayanus, there's been a lot of other strains been discovered around the globe. So we've been working a lot, for example, with this New Zealand isolate of Saccharomyces eubayanus, and that we have sort of an agreement with the discoverers that we can commercialize the strains. So yeah, if anyone is interested in these strains, lager strains that we've created, then we'll be happy to share them.
All right. Maybe there could be some future discussion about some of the stuff.
Yeah.
we're big fans of lagers over here, and recognizing that a lot of these yeasts are very similar to each other. So there's tons of opportunity and tons of room to sort of paint with new colors, so to speak, when it comes to lager.
Yeah.
So I think there is a lot of potential in this stuff in the future.
Mm. Yeah. Mm.
Great. Yeah, actually, that's a good question someone just asked. Is that problematic, the ability to patent and have exclusive rights on a fundamental yeast strain?
Yeah, it's a difficult question, and I know that it's more recently. I mean, a long time ago, 50 years ago. I mean, people weren't really thinking about these things, that they were going around the globe and collecting yeast and using them in their research and commercializing them. But now there's this thing called the Nagoya Protocol. So it's sort of when you take or discover a microorganism in another country, you sort of the original country in which it was discovered in, they sort of have the right to some of the commercial, or they have sort of commercial rights as well. So you can't just, for example, go to if I couldn't go to Canada and find eubayanus there and bring it back to Finland and start making hybrids and selling it. I would have to sort of negotiate with the Canadian government about these commercial licenses and things like that. So, I mean, a lot of people don't at least I imagine that a lot of people aren't even aware of this Nagoya Protocol or don't sort of take it seriously. But at least like the research institutes are quite take it quite seriously. So it's you can't really go. It's a bit. It's a tricky question, but I guess the sort of the thought is good behind that. You can't just go and exploit, especially these. It was because a lot of these microbes were found in these third world countries, so it's sort of to protect them or protect their commercial interests as well. But yeah, it's an interesting topic and not one that I know a lot about, but yeah, I at least know that it's a problem, this licensing of these different strains, or something that has made it a bit tricky to commercialize many of these lab-made strains.
Yeah, it's definitely a challenge, but hopefully hopefully the right thing is done when resources are extracted from one place and commercialized elsewhere, and B, hopefully we're able to accelerate some of that commercialization done in an ethical way.
Mm.
So I think that's going to wrap us up. We've been talking for a little while. I just wanted to say thanks again to Kristoffer for the informative talk and all of the answers. This guy is a fountain of knowledge, and it's always nice talking with him. And we definitely feel very blessed to be working with him every so often on some yeast research projects. And it's really great to have this sort of international dialogue with other brewing scientists. I just wanted to highlight also that tomorrow we're going to be talking about controlling flavor. We're going to have webinars on diacetyl formation, which we covered a little bit today, THP off flavor, as well as Brettanomyces. So a lot of you have been constantly asking us, when are you going to talk about Brett? When are you going to talk about Brett? We're going to be talking about Brett tomorrow. Researcher named Caroline Tarawa, she did her master's thesis studying Brettanomyces fermentation. So a lot of knowledge there. Looking forward to that tomorrow. Thanks again, Kristoffer. Do you have anything else you wanna mention or plug? Any paper?
Not really.
Okay.
Thanks for having me, and I hope most of you were able to follow, even though it was quite a sort of technical and complex topic. But yeah, hope you learned something at least.
Excellent. Well, thank you, Kristoffer, and I hope to see everyone else tomorrow morning or anytime. And also keep in mind that you can watch all of these presentations after the fact on Crowdcast. You can also see them all on YouTube as well. I know Nate has been working hard behind the scenes to get everything uploaded on YouTube today. So you should be able to access everything after the fact. If you want to go back and try to remember how to make a yeast with 100 times the normal amount of banana aroma, you should be able to. All right, thanks again, Kristoffer, and see everyone tomorrow.
Bye-bye.